Very good. Well, good morning, everyone. Thank you guys for joining us on this Saturday. You can really tell it's fall now, right? We got that little bit of crisp air, which I admit kind of love. My name's Liza. I am a research coordinator at CenXL RMCR, and also currently serving as the chapter president of the Rocky Mountain chapter of the Huntington's Disease Society of America, which will henceforth be HDSA, because around here we like to have a lot of really long names. So welcome so much. Thank you guys for coming to our annual Family Education Day. Today's event is presented by the HDSA Centers of Excellence at both the University of Colorado Movement Disorders Center and the Rocky Mountain Movement Disorders Center and Movement Disorders Foundation. So again, now you see all the long names, right? Also want to give a thank you to our organizations that are helping sponsor this event. Without them, of course, we wouldn't be able to be here today, wouldn't have this wonderful food that we will also be able to enjoy. So our sponsors are Genentech, Teva Pharmaceuticals, Neurocrine Biosciences, PTC Therapeutics, Mass Mutual Special Care, Picnic Health, and Unicure. Some of our wonderful sponsors have tables set out back, so please, we'll have a break today, and there will be time during lunch. Please go visit, say hi, see what they're doing for the HD community. Also, I would like to mention, we're going to be at some point passing out some evaluations for your time today. So we'd really appreciate if you could fill those out. Let us know what you liked, what you didn't like, what topics you'd be interested in hearing. We're always trying to improve because today is about you. So we want to make sure that we're taking your thoughts into account. So we'll get those handed out, and then you can drop them off at the CenXL table, which is just the first table outside with kind of that white blue tablecloth. So today, we're going to kick off our presentations with both a familiar face and a new figure in the community. So first, we'll have Dr. Emily Forbes, who is a fellowship-trained movement disorders neurologist at the University of Colorado Anschutz Medical Campus and the director of the HDSA Center of Excellence at the university. She will be accompanied by Dr. Megan Salinas, who's a movement disorder specialist who practiced for several years down in Texas before we stole her away to join Dr. Kumar's practice. And at Dr. Kumar's practice, she will be both very involved in providing clinical care for HD patients, as well as helping with the research studies that are conducted there. Dr. Forbes and Salinas will be presenting this morning on HD research and promising new therapies. So let's welcome up Dr. Forbes to start.
All right, we're working on getting the blinds down a little bit so you can see the slides a little bit better. All right, so of all the years that I've been giving this talk, I think this year I am the most optimistic. 2024 has been a really exciting year for HD research, hence my title, Progress and Hope, when we talk about advances. So I'm really excited to get started. And the outline of today's talk, we'll talk about a couple of studies that are being done here at the University of Colorado, including a new one on HD sleep and cognition. We'll talk a little bit about a survey study we're doing on uncertainty in HD, and then another one on neurologic music therapy, which some of you have heard about before. And then we'll move on to some genetic therapies. And so first we'll talk about what genetic therapies are, how do they work, and then what research is being done. And so when we talk about the genetic therapies, one thing to remember is those are disease-modifying therapies. So those are ones where we're trying to slow down or stop the progression of HD. So we'll start with sleep, cognition, and HD. So sleep affects brain health. And I think we've all experienced this when we've gotten a really poor night's sleep. And then the next day we just feel not ourselves and not right or not thinking clearly. And so we know that just from experience. And we also know that from research studies. So sleep dysfunction has been associated with cognitive impairment in both healthy adults and in people with neurodegenerative conditions. In other neurologic conditions like Parkinson's and Alzheimer's, this has been studied, and they found that in Parkinson's disease, increased sleep is associated with better cognitive performance, and in Alzheimer's disease, or people with Alzheimer's disease have decreased sleep activity. And there's a little asterisk by the sleep because there are different phases of sleep, and we think that there's one that potentially could be more protective than others called slow wave sleep or deep sleep. So the question has been, does sleep dysfunction or having poor sleep promote deposition of abnormal proteins like we see in Huntington's disease and other neurologic diseases like Parkinson's and Alzheimer's? And so whereas other neurologic diseases, this has been studied, this hasn't been really looked at in Huntington's. But it really should be, because we all know that sleep dysfunction is common in HD. It's self-reported in 88% of people with HD that they have some sleep impairment. It's been shown to be associated with cognitive impairment, mood disorders, and worse quality of life in people with HD and also their care partners. And sleep dysfunction can occur prior to disease onset and worsens as HD progresses. But despite the high prevalence of sleep dysfunction in HD, there are a few studies that use what we call polysomnography or sleep study, which is when we bring someone into a sleep lab and we hook them up to measure their brain waves and see what their brains are doing during sleep. So that's what this proposed new study will do. So we're looking for people with very early symptoms of Huntington's disease who just really started to show symptoms. And participants will complete a thinking and memory test and then a sleep study. So we'll come hang out in our sleep lab here at the university for a night. And then the next morning, we'll have them get an MRI. And that's the whole study for patient participation. And from the information we'll get, we'll try to determine if the amount of protective sleep is associated with cognitive functioning. And part of the reason this is important is because there are ways that we can promote or enhance protective sleep to see if that will help people with HD, specifically cognition in HD, but potentially if this shows good results in other areas of HD or other symptoms that HD affects. So if anyone's interested in hearing more about this, I'm happy to talk to you during a break, or we have Lucas in the back who's at the movement disorder table who can talk to you a little bit more about it as well. The next study that we're going to look at is uncertainty in decision-making in Huntington's. So this is one that involves phone interviews with people with symptoms of Huntington's disease or their care partners. And so if you're somebody who has HD and you want to participate, but your care partner does not, that is okay. You can still do the phone interview. And the goal of this is to describe challenges in decision-making for people who have HD and their care partners throughout different phases of the condition. And so we'll use the information to identify ways to make decision-making easier for people with HD. We estimate the phone interview might take up to 45 minutes and people will be compensated for their time. And the last study I'm going to talk about that's going on here at the university is a study in music therapy and fine motor movements in Huntington's. So there's a whole area of research that's dedicated to understanding how music affects the brain. This study entails an initial visit where people come in for a 3 to 4 hour in-person visit, and they'll get some cognitive testing. They'll get the Unified Huntington's Disease Rating Scale, which is when we have people do the finger taps and the eye movements and walking. And then they'll get a specialized brain scan. And then they'll have 15 visits of neurologic music therapy. And those visits can be done at home. So people don't have to come into that. We can have a music therapist set this up at someone's house, and then they can just do the visits virtually. And at the end of the study, we will bring them back into clinic, repeat all the assessments, and see if there's a difference based on the music therapy intervention. And what we're really looking for here is, can this improve fine motor movements? And so fine motor movements are things like tying your shoes, buttoning buttons, sometimes using iPhones or texting. And so we have a specialized way to test this where we have people do a uniform fine motor movement called a groove pegboard. So people have really enjoyed this study. And if anyone's interested, again, Lucas is in the back, or you're welcome to talk to me during one of the breaks. We're still looking for people to participate here. All right, so let's move on to gene therapy in HD. So the rest of the talk, I'm going to talk about studies that are all looking for a disease-modifying treatment, so something to slow down or stop the progression of HD. So gene therapy is a technique that uses genetic material to treat or prevent a disease. And in order to understand the gene therapy trials, we're going to spend a few minutes reviewing the basics of genetics and genetics of Huntington's. So all of our cells have a complete set of all our genetic information. The genetic information is like a blueprint that goes to make proteins which carry out the function of our cells. But not all of our cells will use every single gene. So for example, our brain cells will use different genes than our liver cells might use because they just need different proteins to work. So we have a complete copy in all of our cells, and in different cells, different genes are called up to make the proteins, and the proteins go on to allow the cell to function properly. And the way it's stored And the way it's used is the DNA that's stored gets turned into a messenger RNA. So it's a different chemical. And then that messenger RNA provides instructions to make a protein. And so it's a little simplified version. So we're going to talk about genetics of HD a little bit more. So every case of Huntington's disease is caused by the exact same thing. A gene, the HTT gene, has extra copies of CAG repeats, so these 3 chemicals that make up DNA. And this expansion leads to making a protein that is longer and stickier than it's supposed to be. And so you can see on the image on the left here that the top image, there's a normal size protein. So that's the red in the protein section. I don't have a pointer, but you can see on the top there in the red in the protein section, that's a normal size protein. When the CAG repeats are expanded, that protein becomes really long. And when it becomes long, it becomes sticky, and those proteins find each other and stick together and they clump up. And the way I talk about this a lot when I'm in clinic is it's kind of like when you have like a messy house. So oh, great. So when these proteins clump up, they aggregate and they're everywhere. So when our houses or our rooms get messy, we can always clean them up. But it might lead to us losing things or losing our keys or not knowing where things are, or tripping over things. And so it's good we can clean them up. Our cells don't have a good way to clean up all these proteins. And so what happens with a cell when all of this aggregates, all these proteins clump up, it's really bad for the cell and the cell dies off. And you can see that in this picture on the left. So you have the normal CAG repeats and then the expanded repeats. And the normal protein is made by the normal repeat. And then on the left side, you see the expanded protein, which clumps up. And on the bottom, you can see all those aggregates. And this happens first and foremost in the deep part of the brain called the striatum. And it wreaks havoc on the cells. And so those cells die off. And this is what really leads to a lot of the symptoms of Huntington's disease. And so this brings up the question, how do we reduce that abnormal protein? So how do we prevent that protein from being made and wreaking havoc on our cells? And so this is where the genetic-based therapies come in. So we're going to review 3 broad ways that we can reduce the abnormal protein and prevent it from aggregating. And so in order to do this, I'm going to show you that DNA to messenger to protein again, but I'm going to show you an image that's a little bit more in depth. So I know there's a lot going on this slide. We're going to take this step by step and look at different targets. And the reason this is so complex is because all of these areas represent targets where we can try to decrease the abnormal Huntington protein. So let's start at the very beginning. So this is just our DNA in our genes. So there are some genetic therapies which can directly edit the makeup of our DNA. This includes CRISPR, and the ZFP stands for zinc finger protein. So CRISPR, I think people have heard about because it's been in the news a lot recently. So recently, the first CRISPR technology for a disease was FDA approved, which is really exciting. It's for a blood disorder, which is very different from a neurologic disorder, but it shows that this technology has come really far in a pretty quick time. And so there's a lot of preclinical work that's being done, meaning not in humans, but preclinical before anything's being tested in humans, to see if CRISPR technology can be helpful for people with HD. And then as for zinc finger proteins, there's also preclinical research going on there, but we're not going to spend a lot of time here. Okay, so this is the next step. So we had our DNA, and now you can see in the pink, this is when it's turned into the messenger. So the DNA in the nucleus on the left is transcribed into RNA, and it's called a messenger RNA because it brings a message of how to make a protein out into the cell. This is a really good place to try to block the message so it does not become an abnormal and harmful Huntington protein. So we're going to look at this for a few therapies. And you can see on the left, it says ASO, small molecules, and then we'll put in a little virus on top of it because those are the 3 we're going to talk about. The reason we are going to talk about them because if we can stop that messenger, we can stop that protein from ever being made. And so let's start with talking about ASOs, the first of the 3. So an ASO stands for antisense oligonucleotide. How do these work? So like we talked about, when a piece of DNA is needed to make a protein, the area of DNA gets transcribed into the messenger RNA, and that brings the message outside of the nucleus to where the proteins are made. And so if there's a piece of messenger RNA traveling, it's very vulnerable. If we send in a little piece of nucleic acid, like you can see here in red, it binds to that messenger RNA, and our cells know that that is abnormal, and the cell will just use its own inherent machinery to destroy it. This tactic is being used to test potential treatments for many different neurologic diseases and is used currently for other types of neurologic diseases. So for Huntington's disease purposes, there are two broad types of antisense oligonucleotides. So, and it's based on which one of these the ASO targets. So there are ASOs which only target the abnormal Huntington messenger, so that the normal Huntington gene still gets turned into a protein. This way, the normal Huntington gene or protein can go on and do its normal functioning. The other type of ASO, which is more common, is one that indiscriminately targets both the normal and the expanded messenger RNA. And there are trials being done in both. Before we talk about those two trials, I want to give everybody some notice that the way that we get the ASOs into the area where they need to be to decrease the abnormal Huntington protein is something called an intrathecal injection. So what that means is essentially that it's very hard to get medications into the brain. So our brain has something called a blood-brain barrier. So when we take medications, those medications don't always diffuse into the space that our brain is. And so it can be difficult to get treatments there. And so the way that these ASOs are getting into the brain space is through a needle, like done with a lumbar puncture or an epidural. So you put a needle in the lower spine and inject the therapy in to the cerebral spinal fluid, and then it kind of diffuses throughout the brain. So the first trial is the Generation HD2. So this is tominersen. So I think a lot of us remember a few years ago, tominersen was being tested and the trial stopped because there was some concern that the people who were getting the higher doses were not doing as well, even as the placebo. And so that trial was stopped, but when they went back and they looked at the data, they found that there was a subgroup of people who were a little bit younger and had very early disease onset who actually did well. And so they started another clinical trial just in that group to see if tominersen works. So this is a phase 2 study. It's a placebo, which essentially means it's a sugar pill or doesn't have any medication in it, versus 60 milligrams versus 100 milligrams. The key eligibility for this trial is people should be 25 to 50 years old and have prodromal or very, very early stage HD. And the goals of this are to evaluate for side effects, to see if the amount of mutant Huntington protein is decreased in the spinal fluid, see if there are any changes on imaging in the brain, and changes in function to see if there's any difference in, for example, the ability to move, think, or perform daily activities. And so if anyone's interested in this trial, I think Rocky Mountain Movement Disorder Center is still recruiting for this. So you can you can talk with Liza in the back, who's raising her hand right there, to get more information about this. The update that we got this year for this one was in August. There's an independent data safety monitoring committee. So clinical trials do this where they have just an independent committee that's separate from the trial that looks at safety data. And so that trial recommended that that data safety monitoring committee recommended the trial continue unchanged after reviewing safety information. So that's really good news. All right, so this is the second type of ASO. So this type of antisense oligonucleotide selectively reduces the mutant Huntington protein, or the mRNA, so that the protein's decreased. And so you can see that the DNA on the image on the left, there's the healthy allele, meaning the healthy version of the DNA, and then on the left side, there's the expanded version, so it only decreases the expanded version. This is just the first time that they've shown that an ASO can do this. It's a phase 1b/2a multi-site trial. It's being done outside the United States. It's placebo-controlled, double-blind, meaning there's a non-drug given to a group of people, and neither the patients nor the providers know which one they're getting. So data from 23 participants with early HD has been reviewed. We talked about the administration via the lumbar puncture. The primary outcome is always going to be safety and tolerability for earlier phase trials. And the study period results have been released for 28 weeks. One thing to note about this before we talk about the results is that in order to only target that mutant expanded mRNA, they found that there's a little signal on that mutant RNA or the mutant messenger RNA that about 40% of people have. And so this treatment, as it goes forward, would work in people who have that signal, but not everyone has that signal. So that's just something to keep in mind as we talk about the trial. So in June 2024, they came out with data showing that there was a 46% reduction in the mutant Huntingtin protein in the cerebrospinal fluid compared to the placebo. So compared to the people who weren't getting the medication, they showed that the mutant Huntingtin is decreased. And so you can see that on the graph on the left. And so the blue line shows the decrease in the mutant Huntingtin in people who were exposed to this ASO. And the gray line is people who did not get it. They just got a placebo. And on the bottom arrows, those in blue, that's the points at which people got the lumbar puncture or the intrathecal injection. They found also that there was preservation of the wild type or normal Huntingtin protein. So they knocked down the abnormal protein while preserving the normal one. It was generally safe and well tolerated. And there was also some data that showed that possible loss of brain structures was preserved. So again, this is the first ever clinical trial that demonstrated an allele selective silencing, meaning it's only silencing the abnormal Huntingtin protein. They released additional data. So this is a different graph on the left. This is the total motor score. And they're seeing in the small group of patients that the total motor score is actually better in the people who received the medication versus the placebo. So again, it's early results with a small number of people, but really optimistic. So we're very cautiously optimistic about this. All right, so let's move on. We're still at this point. Yes. Yeah. That's something that the trial study tests for before they select people. So if something like that were to become available, then we would have to test people for this special signal. Yeah. I think they did it by a blood test, yes. Sure. All right, so now let's focus on something called splicing and small molecules. So what this means is we can provide a drug that alters the instructions to make a protein by changing the code. And so how do we do this? So in the image on the left shows that during the processing of that messenger RNA, an additional piece of genetic information is inserted. And so that's the orange. That's the medication. Additional piece of genetic information is put in there. And when it does that, it essentially tells the cell to just destroy that messenger RNA. And that prevents the abnormal Huntingtin protein from ever being made. And so that's something that they're looking at in another trial called Pivot HD. This is a phase 2 trial of a molecule, and the study aims to determine the safety and tolerability at any indication that this reduces the level of Huntingtin protein. And so Pivot HD also came out with some interim results this summer, and they showed that people who took this had a dose-dependent lowering of the Huntingtin mRNA, so the messenger, and protein levels in the blood. It was well tolerated, so there were no serious adverse events. It did demonstrate that it got into the CSF, so that's a big thing in treatments for neurologic conditions. We want those medications to get into the cerebral spinal fluid. And then they showed that something called NFL levels were stable. So NFL it stands for neurofilament light chain. It's essentially a distress signal that the brain sends out. So we have this for different organs. So if somebody thinks they're having trouble with their heart, we can check for a heart distress signal. They think that NFL is a brain distress signal. So they're starting to watch this in a lot of different clinical trials. And so you might hear companies report on this. And so what they showed is that PTC 518 treatment results in lowering of the mutant Huntingtin protein in the cerebral spinal fluid at 12 months. And so what the graph shows is On the left, you can see the placebo. So lowering of the mutant huntingtin protein occurred by about 9%. With the low dose of this treatment, it reduced by about 21%. And with the high dose, it was reduced by 43%. And so that's really exciting news. It also demonstrated a trend of a dose-dependent improvement in the motor score. So whenever, I'm not sure if anyone here has done a clinical trial, I think most people have been evaluated. We make you do the finger taps, we'll make people move their eyes around, we'll make them walk, we'll try to pull them backwards. So that's what they mean when they're talking about the motor score. And so the lower it is on this graph, the worse the motor score is. And they showed that the people who are getting the placebo had a worse motor score than people who were not. And then a few other data points they released. They released the UHDRS, so the full score. So the worsening, again, the lower you are, the worse it is. And people who were receiving treatment did better than the people who are on the placebo. And the total functional capacity. So function is something that we can improve the motor score, we can improve various other measures. We want to make sure people are still functioning well in their life. That's a key measure. And so they showed that people who were taking the treatment did better than people in the placebo from a functional capacity, which is great. So they have said that they are beginning preparations for a phase 3 clinical trial. Again, really exciting news. And so with that, we're going to move on to the third way that we can prevent huntingtin protein from being made. And that means we're going to look at the viruses. So this is a phase 1/2 clinical trial of a product called AMT-130. So it's a gene therapy that's delivered directly into striatum, so that deep part of the brain. And the goal, again, is to slow disease or prevent it from progressing by interfering with the production of protein. So this is delivered by a one-time neurosurgical procedure. So in order to get this therapy to work, we have to have access to the DNA deep within our brain. And so it's a hard place to get to. And we've been talking a little bit about that, about how it's hard for our drugs to get into the spinal fluid and into the brain. And so we're going to talk about how treatments can get into this area. So first we'll talk about how this drug can get into our cells, and then we'll talk about how it can help decrease the mutant huntingtin. So there are two main components of AMT-130. The first is something called a viral vector. So viruses are able to infect us because they have this shell, which you can see here on the image on the left. And the shell tricks our cells to letting it in. And a normal virus that might infect humans will trick the cell to letting it in, and then on the inside of the virus, it has all these viral particles that have our cells' machinery replicate it. However, not all viruses cause problems for humans. There are a lot of viruses out there that we interact with that we just don't really acknowledge or know that much about because they don't really infect us. And so adeno-associated virus is one of the viruses that is not a problem for humans. And so what scientists are doing is they're taking the shell from that virus and removing the interior. So they're removing all those viral particles from the middle and they're putting in a therapy. So what they're putting in the middle is something called microRNA. So small pieces of genetic material that can prevent the production of an abnormal huntingtin protein. And ideally, the cells would use their own machinery to keep producing this for a long time, potentially even for life. But that obviously has to be evaluated as clinical trials go on. So how do we get the therapy into the striatum, that deep part of the brain? Well, that's another tricky process. So it's a one-time neurosurgical procedure. There's a little hole that's drilled into the brain and a tiny catheter is put in to infuse the therapy. So there are two trials ongoing for this treatment, HG-GeneTRX1 and 2. So 39 participants have received either a high dose or a low dose or a sham surgery. So there's some placebo controlled here. We've hit the 2-year mark. 12 people have received the low dose and 9 people have received the high dose. And the data that was released this summer showed that AMT-130 high dose significantly reduced disease progression by 80% using the UHDRS at 2 years out compared to natural history. So one thing to note is this doesn't have its own group of placebo. It compares it to people who have done, for example, Enroll HD or Prevent HD. So we know what the natural history, the normal progression of HD is like. And so it also showed that there's a difference between a low and high dose of AMT 130. And so in the image on the right, you can see that there's disease worsening the lower it is. And so all the way on the left, that's the external controls. So that's the people who have HD, who have been in other studies where we just monitor them over time. And there's improvement in people who are in low and high dose of this therapy. And so another really exciting development. So with that, I want to thank everyone for coming this morning. And I wanted to give a big thank you to everyone who has participated in a clinical trial and everyone who has supported someone who's participated in a clinical trial. I think it's a very brave thing to do, and it shows a lot of commitment. And I think the talks that we have this year really show the breakthroughs that we've had because of the dedication of people who've participated. So thank you so much, everyone, for being here and for participating in trials.